The last of the biochemical molecules we have to talk about, all right? Or their name for polypeptides is a protein. And they're basically just large macromolecules made of chains of amino acids, and then they fold on top of each other. All right, so they fold on top, fold into these really weird shapes. You saw that with the video. Again, those of you watching at home, you should watch the end of the video. Hold on a second. Spear you with it. You should watch the end of the video of the Crash Course Hank Green one, because it talks about a lot of the stuff I'm going to talk about today. Although I'm going to go into a lot more detail. I mean, so much detail that it's not going to be able to get done in one period. We're going to split it up into two. I'm going to do half and half. I have a lot of little video clips to show you how this stuff goes on. Some of them are going to show you in real time, others breaking it down, and still others summarizing it at the very end. All right, so that's what they are, amino acids. They do everything in your body. That's the whole point of them, okay? They basically are your building materials. They are enzymes. They make you what you are. They give you your color, your eye color, your hair color. They digest food for you. They they make you cough. Sorry, that wasn't on purpose. They do everything. And this one's got to work too. All right. So eventually you're going to make that proteins. But how do we get from DNA, which we talked about yesterday, to actual proteins? That's the point. That's what I want to go over today. It's not so easy. I think I said this before. I think most biology teachers would agree. I'm a chemistry teacher, but I think most biology teachers would agree. There are only a handful of topics in high school biology that are difficult to actually understand. Most of the high school biology, classification, cell bio, even ecology, is basically a matter of you learn something, you memorize it, and you spit it back. It's not that hard. But when you talk about the Krebs cycle, the light and dark reactions, photosynthesis, and transcription and translation, those are the topics that are actually hard to understand. All right, they're really not something you can just memorize and spit back. So you kind of have to know what's going on in each of these cases. And that's why I'm going to spend some time on this. I think I'm going to have a little video clip right now. Hold on a second. Is that coming up now? No, one more line here. So amino acids are linked by the peptide bond. That's right. I got to show you what an actual. I have one more picture here to show you here. I got to show you this actual picture of what's going on at the chemistry level. Actually, you're going to understand this picture a lot better if you take Chem 2. I told you I moved this unit down from Chem 2, which is really kind of where it belongs, but I couldn't do it anymore because we turned Chem 2 in AP Chem, and there just isn't any biochem in AP Chem. There's very little organic chem in the AP exam. So I kind of had to get rid of some stuff from that class. And this is one of the things that I didn't want to throw out completely, but you'll understand better if you learn, for example, in organic, you're going to learn that this is a functional group called a carboxylic acid. Probably should use red for that because I kind of think of acids when you see red, don't you? Because of litmus. This is a functional group called an amine group. All right, so that's the amine group, that's the acid group, hence the name amino acid. The amine group is actually basic, and the acid group is actually acidic. Oh, use red for that. So it's like an acid-base reaction. Say, okay, well, that's nice. It's like an acid-base reaction, only instead of having an inorganic acid base like HCl and NaOH, you actually have an amine and an organic acid with a carbon group here. The big difference between this guy and an inorganic one would be the carbon. That's the B. There's a carbon there, there's a carbon there. It's got carbon in it. So if it's got carbon in it, it's organic. All right. So again, the second part about this, that's the actual reaction that's going on, and it splits off water, as you can see. But here's the other part of it. It can do it on both sides. I can have this guy continue to grow that way, and this guy continue to grow that way. So I can keep adding more amino acids. And I got different ones, because remember, there are 20. He said that in the thing. Some guys have just one carbon in them. Some guys have multiple carbons. Some guys have other things in them. You'll see some pictures of them, all right, on a handout I'm going to give you. You don't have to memorize it. But the point is, this can grow into a long chain. As these guys get hooked, as that carbon gets hooked up to that nitrogen, or across the oxygen there, you can get a long chain of these guys. And that chain is what makes hemoglobin. Let's just use one that you've heard of, which carries oxygen, through your blood. It makes insulin. It makes everything that makes you, basically. Okay? All right. Now I got a little video clip. Now I'm going to show you this clip, which is going to be the full thing happening, transcription, translation, all happening, making proteins, described for you in real time. This is the crazy part about it. This is going on in about a couple trillion cells in your body right now as we speak, and it's happening this fast. It's pretty amazing. Like a factory could not do this. All right, hit that light for me, and we'll watch this little video clip here.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U.
Uracil.
You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
100 trillion of those per second in your cells, basically to keep you alive. That's how much blood you're making all the time.
How do they know that? They don't.
Don't ask me. Hit that like for me. All right, look, big picture first of all. Let's get a big picture, and then then I'm going to basically condense it down to each of these different steps. Big picture is this: your DNA is like a blueprint that you're building a house, but in this case, your house is you. Okay, and the blueprints will be large. They'll contain every single room in the house, all the plumbing, all the wiring, the roof, everything, the foundation. And now, not every single person who's working needs to know every single thing about it. All right, so say, for example, I'm just building the one room. I'm just doing the plumbing, or I'm just doing the wiring, whatever it is I'm doing. Well, I'll get those pages from the blueprint. I'll read those off, and I will make that just that one thing. I'll put my pipes here for this room. I'll make my living room. I'll do my brick fireplace, whatever it is. I get that page of the blueprints, and I read it off, and that's what I do. Well, that's exactly what you're doing here. You're basically reading off that page of the blueprints, sending it out to the subcontractor, saying, here, read this and produce my fireplace for the house. And that fireplace, of course, in this case they made a second ago, was hemoglobin. That's one of the things that help your blood carry oxygen, okay? All right, so that's the big picture. Now, how does this happen? That's what I got to talk about now, how all of this actually gets done, all right? Protein synthesis. By the way, you're going to notice something about your papers. I have an extra slide on there. You're not actually writing these notes at the top of your paper. Just ignore that. You can cross that all out. I actually put an extra slide in there. You are halfway down the paper now, okay? And that's where this is going to go. Under V5, protein synthesis, DNA is the ultimate source of all information in the cell, like I said before. Okay? Like I just said, it's the blueprint for the whole house. And you're way more complicated than a house, people, although some of you aren't much brighter than a brick wall. See, there was one of my jokes. Look, you're a lot more, if you think about that, trillions of times per second, that's what's going on in just one of the types of cells in your body. Every cell in your body is doing that. You got trillions of cells. It's just incredibly complicated. You are, all of the factories in all of the world, and all of the cars, and all the complexity of everything in the world doesn't come close to you. You are way more complex than pretty much anything we've made. We often worry about, I was thinking this the other day, we're worried about, robots taking over the earth and artificial intelligence and crap like that. Self-driving cars is a new thing now. And we're so worried about these kind of things. And we're so impressed when our computers can do something that we can't do. You got to remember something. We made the computer. And while making the computer, we were keeping ourselves alive. All right, that and alone, just keeping you breathing is more complex than all the computers ever made put together. I mean, it is an amazing thing. We sometimes don't think of that. We think machines are going to sometimes replace us or better than us. I mean, there will be jobs lost because of it, but I'm just saying, I don't think you have to worry about the robot apocalypse right now. Maybe the zombie apocalypse once the Zika virus gets us, but I don't know. We'll worry about something. All right, so DNA is the ultimate source of all information. And of course, what happens to this information? It's used by the cell to produce proteins. You are proteins. You are what you are. Basically, that's what you are. It's proteins. And basically, these proteins are made in a two-step fashion. Two-part process. Now, I have some of these notes filled in for you. Most of them you're filling in yourself. But imagine, again, you have these notes here. You have all these pictures here. It seems like you're writing a lot today. Imagine how much you would have had to write if you were taking these notes from scratch. Unfortunately, you say, well, why don't you just give them all to us? Because then you don't listen. I've said that before. You go off to college, you will get professors who will put their notes online, who will give you their printed out PowerPoints, and you'll decide that I don't need to listen because all the information's here. Because hey, there's a lot of information on my computer. There's a ton of information on any Google search I do, but it doesn't mean I have that information. All right, I have to actually know it, and I don't if you're not taking those notes on this. So anyway, it's a two-part process, and the first part is called, you have this down, transcription. And that's already filled in for you. What is transcription? That's the part you saw happening first. We're only going to talk about transcription today because we won't have time to do both. Okay? We're just going to do transcription. I will give you you will not be able to finish the worksheet until you get the second part of that. So, but I'll give it to you to start probably at the end of the period, but you'll have to wait till tomorrow to finish it because you won't have all the information yet. So transcription is the process by which information contained in the genes, that's just a segment of DNA, is going to be copied to make messenger RNA. Okay, copied to make messenger RNA. That is like the runner. Okay, here's my blueprint. I'm the architect, and I give my runner, I give my secretary, I give somebody, here, take this page of the blueprints to my mason. He's got to lay the bricks for this. Okay, that messenger RNA is the runner. He's the secretary. He's the guy running the plans to somebody else. So he's got to make a good copy. He goes, okay, give me the photocopy machine, puts it on the photocopy machine, and he runs it out to them. That's what his job is. And that is done by messenger RNA. There are different kinds of RNA we're going to talk about. It does it by splitting open, as you saw in the little video clip. Splits open the DNA. It reads half of it. By the way, how is it able to read it? It's able to read it because it matches up those adenine, thymine, guanine, and cytosine nucleotides with its complementary one. All right? Only it replaces one guy. Who's the guy that gets replaced in there? Yeah, with what? Thymine gets replaced with uracil. Good. All right, we're going to mention all that and say, oh, I'm back to another video clip. This, we're going to slow down. We're going to slow it down. That first one we saw was real fast, real time. You could not tell what was going on. Now we're going to slow it down, look at it a little bit on the macro level here. Turn that light on. Off of me. And let's see what's happening in the transcription part.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis.
Okay, so that's the part I want to talk about right now, how that messenger RNA gets built. Hit that light again? Okay, here it comes. RNA polymerase, that's the name of the, it's going to be one of the questions in the little worksheet you're going to get in a bit. RNA polymerase is the name of the enzyme that helps to unwind the DNA, form this transcription complex that gets this whole thing going. Gets all of these nucleotides to come in and pair up with their opposite one and link them together to create the messenger RNA. It knows where to start, by the way. Everybody said, and she said in the video clip too, the segment of DNA, she said off to the left or to the right, or whatever way you want to look at it, is the gene is this segment of DNA. Well, how does it know where to start and stop? I mean, there's a lot of letters in this DNA, okay? So how do I know where to start and stop? Just a small portion of it actually corresponds to a specific protein, a specific amino acid sequence. So what it has to do is have a little start codon, a start sequence here, and a stop sequence there. It'll tell him when to start and stop and break off. Okay, so the transcription complex recognizes the start of a gene, and then it unwinds it, and unwinds that segment. The nucleotides come in and pair up with 1/2 of the strand of DNA. And then the same RNA polymerase binds those nucleotides together and makes up the messenger RNA, that thing that's snaked out of the nucleus and went out to make the actual protein. Remember, it can unwind this while you're copying now. Just say it can unwind this fairly easily because the things holding those together, those nucleotide bases together, are just hydrogen bonds. All right, you're not breaking covalent bonds or any kind of ionic bonds. You're breaking hydrogen bonds, which are relatively easy to break compared to those. That's how it's able to unzip. By the way, the same process of unzipping happens when DNA needs to replicate. Okay, but we're not going to talk about that. There, if you're just replicating the DNA, you would just find complementary ones, and they're not going to be a U, it'll be a T again. And you'll make two things right before what? Right before cell divides. Remember that? What were the steps of cell division? Remember those? Yeah, mitosis. What was it? Interphase? Interphase, prophase, metaphase, anaphase, and telophase. Good.
I remember from bio. Okay?
Yeah, you should. You should remember that. Okay. Now,
It was only last year.
a few words about RNA, because although I know I've said this a couple of times, and it's actually on one of your graphics, I never actually wrote down in your notes the differences between RNA and DNA. So I want to make sure I have it on here, okay? It's similar to DNA except for the following things. So you want to write these down. These are the things I'm going to ask you about. How is RNA different? One of the ones is you got to give me some sugar. It's the sugar's different. Yeah. Right. Did I? RNA has a ribose sugar, and of course DNA has deoxyribose. That one we kind of forget about sometimes, even though it's pretty easy. The one we remember the most, I hope, is the uracil part. What does uracil replace? It replaces thymine. And the third thing is, and this is another one we kind of forget about, RNA is a single strand, not a double helix. It's just a single strand. So that's a biggie too. Now, in addition to those differences, I need to talk about the different kinds of RNA you can have. Okay, we actually have, as we've already mentioned briefly, messenger RNA, spent the most time talking about that today. Transfer RNA, we're going to spend the most time talking about that one tomorrow. And there's also ribosomal RNA, which I'm not going to talk about really, but it does serve a function as well in this process. Okay, let's start out with different types of RNA. The messenger RNA, or often abbreviated mRNA. Okay, it stands for messenger RNA, and it basically carries a copy of the genetic information. Like I said, you put on a photocopier, and you run it out to your subcontractor who's doing your electrical wiring. Maybe your electrical wiring is, something in some function of your body to keep you alive. Remember, the analogy of blueprints and a house really breaks down because a house isn't a constantly living thing. Basically, you build a house and it's built. This is happening all the time to keep you going. The eventual goal, remember, is to make a protein, to put those amino acids together and make a protein. The messenger RNA basically his function is to carry a copy from the DNA out into the cell to be translated eventually into a protein. But right, he does that through the translation. He needs help with that. Okay, he's just carrying the copy out from the DNA. Okay, all right. So the transfer RNA is the other kind. This is one we're going to spend more time on tomorrow. So this is my last slide here. I'm not really going to do any more today. Okay, so let's finish up transfer RNA just to tell you what he is, but we're not going to really work with him until tomorrow. Although you've seen it happening with the little video clips. Transfer RNA contains a sequence of 3 nucleotides. That's what it's like. Think of the nucleotides as letters, and then the 3 together that make what's called a codon as words. And then the words put together as a sentence. Okay, if you can think of that. All right, we'll use the analogy of building. Let's use the analogy of building a story. Okay? if I want to write a story, if I want to write a script for a movie, all right, I'm obviously writing with letters, and they're using the letters here would be the individual nucleotide bases. But, you know, obviously we don't really read letters one at a time, we read words. The words would correspond to probably, more likely, be the actual amino acids. I mean, I'm sorry, the codons. All right, and then they transfer a whole bunch of them together, put together into amino acids, makes sentences, and eventually the story itself is you. You are the story. How about that for now? I just made that up. Thank you. Thank you. Yeah.
Are you gonna make us a code?
I am going to have you do a few codes. Not many. That will be not probably partly tomorrow, the next day. We have Friday's a test, so we have like some worksheets to practice that. It's not hard. Oh yeah, back to this. So I'm not giving you. But just real quick on this, to transfer RNA then is again. Look at this guy's made. If you look carefully, can you see it on your things?
Absolutely not.
The little letters. Those little letters correspond to nucleotide bases. Notice there's a U, a C, an A, whatever. And at the bottom, there are 3 that match up against the messenger RNA. We're going to see this tomorrow. And then at the other end, what's at the other end? An amino acid. That amino acid, at this end, the anticodon at that end, this guy matches up to the messenger RNA. We'll see that tomorrow. And these guys all get linked up as they get pulled through like a ticker tape, which you don't even know what that is. I don't even know if that's used anymore for anything. Okay, so tomorrow, by the way, before the actual codes here, code for individual amino acids, like leucine, serine, phenylalanine. All these different ones are coded, sometimes by more than one sequence. UUU, UUC, UUA, UUG will all, those 2 will give you phenylalanine, these 2 will give you leucine, these 4 will give you leucine. All right, so you have more than one possibility to code for these guys. The same amino acid may be coded for by more than one RNA codon. And a codon is just a sequence of 3 nucleotides. Finally for today, one more thing to pop up there. Finally for today, how does it know when to start and stop? Well, you can see there are certain stop codons here, and the start codon is usually AUG, which is methionine. Okay, a start codon. So it knows where to start and where to stop a gene. It can stop at these places. It starts there, AUG. All right, some codons do that. Tell where the RNA polymerase should start unwinding and reading this stuff. And making your protein. All right. Now, I'm going to stop here because tomorrow, we certainly obviously do not have enough time to do translation in the same period. Tomorrow, we will talk about how we go from the messenger RNA we created in the transcription process to the actual amino acid sequence and then eventually the protein.